Hi, let me introduce myself. I'm Miriam.
I'm Patrick.
I'm Iman.
Our topic of choice is plant defense, but first we have to explain what that actually is.
Plants are faced with a variety of bacteria, fungi, and pathogens on a regular basis. In order to make sure the plant doesn't get too damaged, they've developed a series of defense mechanisms. These include two broad categories: mechanical defense and chemical defense.
Before we start talking about the defenses, I would like to give some examples of the mechanical plant defenses that can aid in the protection and resistance for the plant, which include hairs, trichomes, thorns, spines, and thicker leaves, cutins, suberin, and waxes. The defense can either be direct, which is when the plant confronts the herbivore, both directly by affecting host plant preference or survival and reproductive success, and also indirectly through other species such as natural enemies of the insect pest. In this part of the presentation, we will focus on direct plant defense. So some plant structural traits such as leaf surface wax, thorns or trichomes, and cell wall thickness and lignification form the first physical barrier against herbivores and play an important role in host plant resistance to insects. Structural traits such as spines and thorns, trichomes, toughened or hardened leaves, incorporation of granular minerals into plant tissues, and divaricated branching can also be helpful. First, lignin, a phenolic heteropolymer, plays a central role in plant defense against insects and pathogens. It limits the entry of pathogens by physically blocking or increasing the leaf toughness by reducing the feeding by herbivores, and also decreases the nutritional content of the leaf. Lignin synthesis is induced by herbivore or pathogen attack, and its rapid deposition reduces further growth of the pathogen or herbivore fecundity. Second, trichomes tend to be more effective against insects that are small relative to trichome size. Additionally, trichomes tend to deter sap feeding or leaf chewing insects to a greater extent than those feeding within the plant tissue. They interfere with the movement of the insects and on plant surface, thereby reducing their access to leaf epidermis. It is also noted that the trichome density changes depending on the herbivory attack. When the plant is being attacked, it increases in density to protect itself from the insects. Third, the basis for the toughness of plant tissue in the cell wall is also known as fiber for nutritionists. It is a mechanical composite which is organized as a mixture of two or more components, which help to protect plant in certain areas.
First, we have to understand that the main form of chemical defense in plants relies on secondary metabolites. These are organic compounds not formed for development or basic growth, but for defense against predators. Within this, there are four broad categories: phenolic compounds, terpenes, alkaloids, and cyanogenic glucosides. Let's start with phenolic compounds. These are mainly derived from aromatic amino acids, which include phenylalanine, tryptophan, and tyrosine. Keep in mind that this group is the most diverse group of secondary metabolites in plants. Phenolics can also include other compounds, such as flavonoids and tannin. Tannin can be used to make plant tissues indigestible by herbivores, making it less likely that herbivores will eat the plant. Next is terpenes. Terpenes are made by the MVA mevalonate pathway and the MEP methyl erythritol pathway with glycolysis. The main function of terpenes is to act as an organic volatile compound against herbivores. One of the most understood ways it does this is by inhibiting acetylcholine esterase in mammals and insects, ultimately terminating nerve impulses. One terpene that does this is limonene, which can act as a competitive inhibitor of the acetylcholine esterase, or as an uncompetitive inhibitor, which means that it will bind to the enzyme substrate complex, leading to its inhibition. For competitive inhibitors in terpenes, they will bind to the acetylcholine esterase and prevent acetylcholine from binding, thus blocking the neuromuscular junction. Uncompetitive inhibitors will bind to the enzyme when it is bound to the substrate to allosterically alter its shape and prevent product formation. Terpenes can also act to increase the inhibitory neurotransmitter GABA by allowing more influx of chlorine into neurons, which can prevent action potentials. The third category of secondary metabolites is cyanogenic glucosides. They are derived from aromatic or branched amino acids. These metabolites act as deterrents so herbivores will not eat the plant. But keep in mind that these compounds can also pose a possible threat to the plant itself, examples including plants producing less flowers. And these metabolites are very energetically costly to make. This is since an energy-rich molecule, NADPH, has to be used more than once to make this metabolite. Typically, cyanogenic glucosides are produced after part of a plant was attacked. They will be hydrolyzed by lyases and other enzymes, ultimately producing hydrogen cyanide, which acts as a toxin that can bind to hemoglobin and inhibit respiration of the attacker. The last group of secondary metabolites is alkaloids. This group is subdivided into three categories: true alkaloids including nicotine and morphine, pseudoalkaloids like caffeine and capsaicin, and protoalkaloids like mescaline. Proto and true alkaloids are derived from amino acids. Alkaloids' main function is to act as a toxin against herbivores due to disrupting neuronal signaling, interfering with DNA replication, protein synthesis, and enzyme synthesis. They can also affect the concentration and expression of neurotransmitters, which is the function of nicotine and caffeine. Alkaloids like emmetine can cause mutations during DNA replication of the herbivore by directly binding to the nucleotide bases.
Plants have countless defense mechanisms they use to defend themselves from biotic and abiotic stressors, ensuring their survival, growth, and reproduction. We will be learning more about how plant defenses can help with eco-sustainability, understanding evolution, and promoting agriculture, sustainable agriculture that can help with human health. Plants defend themselves from biotic stressors like herbivores, pathogens, and other plant competitors by using chemical defenses like alkaloids or tannins to deter herbivory, or phytoalexins to block pathogens. They can also develop physical barriers like waxy cuticles, thorns, trichomes, or lignified cell walls. Some release allelopathic chemicals to suppress nearby plant growth. To survive abiotic stressors like these, they can produce antioxidants or alter metabolic pathways, like changing their biochemistry to suit their needs. Plant defenses drive evolution, ensuring survival through herbivory control and reproduction. In agriculture, they can help promote pest-resistant crops via genetic engineering and selective breeding. By reducing pesticide use, they promote sustainability, fostering eco-friendly farming and food security. This is kind of an intersection where food sustainability meets agriculture meets evolution. Let's talk about the future of plant defense with epigenetics and CRISPR. Epigenetics helps plants adapt to stress through DNA methylation, regulating defenses without genetic changes. CRISPR-Cas9 enhances this by modulating genes without cutting DNA, providing a reversible, sustainable alternative to GMOs. These innovations boost crop resilience and sustainable agriculture.
That's all, folks.